Welcome to today's webinar. Peering Beyond the Chapter, Elevating Sterile Compounding Practices is presented by Dr. George Smith, System Sterile Compounding Services Specialist with Prisma Health Department of Pharmacy. Dr. Smith is a board-certified sterile compounding safety advocate. In addition to his position at Prisma Health, he is also a clinical instructor of sterile compounding for the Kennedy Pharmacy Innovation Center through the University of South Carolina College of Pharmacy, and the principal of Echelon Compounding, a consulting firm dedicated to advancing sterile compounding and technology. Dr. Smith's career has traversed many areas of pharmacy, including retail pharmacy management, pharmacy legal regulatory inspections, home infusion partnership, and multiple clinical leadership roles within health system pharmacy. Dr. Smith has dedicated the better part of his career to promoting safe practices for medication delivery and raising awareness of the increased risk involved in sterile medication compounding processes. Today's webinar is ACPE accredited for 1 contact hour. To receive CE credit, complete the evaluation link on the last slide of the handout found in the GoToWebinar toolbox under Handouts. We are recording this session and will share the recording and evaluation link in a follow-up email. You can submit questions during the webinar using the GoToWebinar toolbox under the Questions tab. We will answer the questions submitted following the presentation. Thank you for joining today's session.
Thank you, Amy, and thank you to ARL Biopharma for hosting this webinar. Let's go ahead and get started. I have nothing to disclose, the legal regulatory part of this. And also, like Amy said, this is available for 1 contact hour of continuing education. So diving right into our learning objectives, we're going to talk a little bit about the alternative technology statement in USP. As you can see, we're diving into some very interesting topics here today. We're going to look at USP monographs for sterile compounding, and we're also going to discuss and identify the factors that contribute to the determination of when to protect a CSP from light. So we're going to start with using today's technology. So our first question is going to be, what is true concerning the application of the utilization of the alternative technology statement contained in Section 1 of USP 797? You know, are those alternative technologies prohibited unless the manufacturer has a statement from USP? Are they not prohibited if the technology has been approved by the FDA? Or what about if the statement has been included in each of the proposed revisions since its inclusion? And finally, the alternative technology statement is meant to encourage innovation. And the second question we'll discuss is all about process validation. And it can be characterized in a number of different ways, and we'll get to that as we go through. So the first thing you may be asking is, what are you talking about, George? What is this alternative technology statement? Well, it's actually right in the beginning of USP 797. And if you're anything like me, I gloss over this part all the time, and I'm just specifically looking for something specific. But here it is, right here in the very first paragraph of 2023 USP 797. So the important concept here in this statement is really talking about proving the non-inferiority and being able to validate any of these technologies that we want to use in addition or alongside USP 797. So what are the basics of the technology statement? Well, it really boils down to that the inclusion of the statement is meant to encourage and not discourage the innovation of technology. It first appeared in the 2008 version, but was conspicuously absent from the other revisions all the way through to the 2022-2023 cycle. USP does not prohibit the use of alternatives provided that these alternatives are demonstrated to be non-inferior as explicitly designed in the chapter. In the validation process of most technology, while vigorous, we must remember that the traditional method is manual compounding. The innovations like automation and robotics are developed to be superior to manual processes, but still must be validated. When we speak of validated or even the word validation, we're actually referring to process validation, which has many similar definitions, but slightly different definitions. As you can tell from this particular definition, process validation does not only validate a specific process, but in and of itself is also a process. Process validation is an ongoing assessment of all the elements in the procedure. While process validation is an integral principle in cGMP, it is not explicitly defined by the FDA as to how to conduct a process validation of cleaning agents in the clean room, for example. The expectation from the FDA is that you have evidence to support that your process meets your predetermined criteria for validation and success. This graphic is fantastic at depicting the overlapping relationship between validation, qualification, and the verification process. It even breaks down the qualification process for us. As you can see, it uses verbs that are in the present tense. Words like verifies, meeting, ensuring, as opposed to using verbs in the past tense to describe something like a snapshot in time. The FDA has some guidance for industry on the principles, just not the how-to. And as pharmacists and compounders, you know, we like directions, instructions, checklists, things like that. But that's not exactly how process validation works. Regrettably, process validation is a very difficult concept for most us pharmacists to actually comprehend. So 2 slides ago, I stated that process validation is complex, and it is. But what we have here is going to be my attempt to simplify the concept by breaking it down into 4 buckets. Performance comparisons. We're comparing against the standard, so the new technology against the standard. Repeatability and reproducibility. Can we deliver similar results across the spectrum of the different conditions? Accuracy and precision. Do results match the true value with minimal variation across the board? And then finally, specificity and sensitivity. How well does the method or the process identify the positive and the negative? Not just the good or the bad, but also the presence versus absence of a value. So now that we've scratched the surface of process validation, here are just a few examples of how the alternative technologies statement can be viewed through the lens of USP 797 in emerging innovation. Rapid microbial methods. We're looking at this versus USP 71, so sterility tests. But we also have in the definition or in the statement itself, USP 1223, talking about the validation of alternative microbial methods. So this is becoming a little bit more mainstream, and with the different labs and different ways that we can do this, alternative microbial methods is becoming a very, very important part of the sterility testing process. Automated visual inspection technology. Well, we're comparing that to the manual standard of USP 1790, visual inspections of injections. There's some really cool stuff out there that not only is much more accurate, it is much faster. If you've ever really done any type of long-term visual inspection of CSPs, it gets really tiring, and it's really hard once you get tired. There's built-in breaks, and it is a very difficult and time-consuming process. Sterile compounding automation, that versus the traditional manual compounding. What does that look like? Innovative container closure systems, such as cyclic olefin copolymer bags and syringes. These new container closure systems, very different than the PVC or some of the non-DHEP bags that we see today. We're talking high clarity and transparency. They are very inert. They do not contain plasticizers, low moisture permeability. Thinking through it from sterilization compatibility of being able to sterilize inside the bag, very leachable, resistant. And actually the most exciting part of this to me would be the possible ability to add UV light resistance incorporated directly into the materials, and we're going to talk a little bit more about protect from light as we go on. And finally, USP 797 batch yield limitations. This may be one of the most popular questions that I receive, and that some of my colleagues and some friends on this compounding expert committee is really, why did the compounding expert committee with USP 797 limit the yield? And secondly, where did that 250 magical number come from? So the question then becomes, right after that, to the people that have really studied that first section of the chapter is, well, can I apply the alternative technology statement to increase my yield to, let's say, 500 or maybe 750 or maybe 1,000? Can I do that? Well, we're going to take a closer look into both sterile compounding, validation qualification, and the information and science behind the batch yield limitations. So let's evaluate sterile compounding automation using my oversimplified example. A validation concept may appear like this: performance comparisons. We're looking at, are the final doses sterile? Are the final doses as accurate as the manual process? I would argue with automation, probably more accurate. Looking at repeatability and reproducibility, again, are we able to produce the sterile doses in the same type of environment as manual compounding? Is this an ISO 5 enclosure? Can the automation reproduce a final dose using different concentrations of a specific medication? So same dose using different concentrations of the medication. Maybe not all at one time, but can we get that reproducibility, accuracy, and precision? Can the automation make an accurate dose with respect to what is requested? We're looking for 1,000 milligrams of cefazolin in a bag. Can we do that? And what is that acceptable variance? What is that? Plus minus 4%, plus minus 5%, plus minus 10%. Additionally, over X number of doses, maybe an entire batch, is the variability within a predetermined acceptable range? And finally, specificity and sensitivity. Are appropriate doses passed or exported for accuracy of the dosing, and are inaccurate doses rejected or kept back? Are they at least marked on, notified that they are rejected doses? That's my oversimplification of really looking at what a validation of sterile compounding automation would look like. But the second part of that, when it comes to the automation, becomes the qualification. We did see earlier that qualification, as you will recall, is a subset of the validation, that design qualification, install qualification, operational qualification, and performance qualification should answer the fundamental question: is the equipment, system, or the process suitable for the intended purpose? That's the question we're trying to answer. Ideally, when you look at a piece of equipment like this or a system, the vendor is going to be responsible for the design qualification, determining is the design appropriate. Then the install qualification, is the automation or the equipment installed correctly according to the manufacturer's specifications? We also call this acceptance. So install qualification is termed acceptance, testing protocol, or just acceptance sometimes. But then on the second phase of that is the automation vendor or the equipment vendor willing to come alongside the customer and assist them in integrating the automation into the compounding operational environment for operational qualification and performance qualification. It's, some of this automation for sterile compounding robotics been around for a number of years, over 10, and we're seemingly getting good track record of these being validated as being non-inferior to manual compounding. Still a long way to go for this technology, but it does seem like the future is bright. So moving from, manual compounding to automation in a lot of senses has proven both to be non-inferior, and in some ways we could say it could be considered superior. So let's shift gears a little bit and talk right about the topic a little bit more controversial, the batch yield limitations found in 2023 USP 797. Specifically, it is under section 12.2, which is titled sterility testing. So USP 797 states that the maximum batch size for all compounded sterile products requiring sterility testing must be limited to 250 final yield units. I'll be honest with you, I am a little surprised that so many have found this concept of limiting the batch size to 250 so problematic in their operations. Sure, making more batches may cost more money, different places around that operationally. But personally, I think manually compounding 250 units as a single batch seems to possess a high level of risk of contamination to me. So I'm often asked two questions when this subject comes up. Why did USP 797 state a limit? And how did they come up with or decide on this low maximum limit for products that undergo sterility testing? And the second part of that is we brought up a second ago, was can we use the alternative technology statement in USP 797 to validate our process inside our pharmacy or inside our facility to increase our maximum yield beyond 250 units? That's the big question. As you would expect, the answer to both those questions is very complex, but I think can be answered by evaluating the available data and process validation concepts. There are two issues that I think should be mentioned to help explain why the Compounding Expert Committee decided on the limitations and help us guide that process validation that would be required. This is a pretty sobering theory that environmental microbiologists estimate that less than 2% of the bacteria can be cultured in the laboratory. So if we use that theory as a guide, a bacterial cell on growth media is not going to give us a CFU every single time. 98%, thinking about that, 98% of the bacteria would be unculturable. And it really becomes, if you look at it from a USP sterility testing and environmental monitoring perspective, it's pretty unsettling to me as a scientist that that's where we are in 2024. The second issue is the sterility test. The compendial sterility test is not a test for product sterility. This is not new, nor it is particularly insightful, according to the authors of the study, but it has been frequently presented as a flawed test for its stated purpose in the literature over the past 80 years. Think about it. We are probably perform at a high enough level that we only contaminate a small percentage of the batch. So how do we find that small percentage that could be contaminated? And more than likely, we are going to pass with a low level of contamination. This is really a landmark statistical analysis here of the probability of failing USP 71 sterility testing. And it really, people much smarter than me did the statistics and really look at how to understand this. But if you think about just this spot right in here, 0.5%, that we're looking at, if we're making let's say a batch of 100, then it probably one could be contaminated, or 0.5%. If we look here, 1 in 100, well, these two numbers tell us that 1 in 100 of a batch, there's only an 18% chance of failing the sterility test. So that there's a great chance that that one that could be contaminated and be very dangerous would pass the sterility test. So this is some of the evidence that the compounding expert committee used to determine that as these batches get bigger, we could contaminate some, and that there's a very good likelihood they would not get picked up on the USP 71 test. And that's concerning. I'm convinced there's enough evidence here to support at least a maximum, and I like the 250 for a typical 503A. But the question still remains, well, that's great, George, but can we still use the alternative technology statement to do this? Well, yes, and it's a can versus should. So let's examine what this validation might look like in this specific example. Let's apply the same process validation logic and example as we did the sterile compounding automation and assess the feasibility and maybe even the appetite for designing the process validation plan. This is kind of the framework, but the old saying here really points this out. The devil is in the details. What does it look like to develop process validation to increase batch yield greater than 250 to prove non-inferiority? It really boils down to a couple different things. Consistently delivering, we must be accurate and precise, and then being able to accurately identify both the positives and the negatives. And that's going to look like something that you're going to understand and you're going to have seen before, but let's look at what it's going to entail. I think it's going to take a lot. So this is just an example for the sake of discussion of truly developing this process validation to increase the batch yield beyond the 250. Now, truly developing a process validation program is beyond the scope of this presentation. However, I broke this down into 3 buckets to say that, these are some of the areas that would contribute to a process validation of this. Let's start with environmental controls and monitoring. What do we see here? What does this need to look like? Well, probably continuous non-viable airborne particle sampling in all classified areas. It's not just every now and then. And we're talking continuous in the PEC, continuous in the SEC, in the room. We're talking really, we must control the environment to a greater extent than the chapter and prove that this state of control is both achieved and maintained. This looks like viable air sampling during each batch, viable air sampling during dynamic conditions multiple times a day, not just once, twice, but multiple times a day, not just every week, throughout all of the class 7 space in the SEC. Surface viable particle sampling during each batch in the PEC. I think you're starting to see this becomes a little difficult. This is like having a whole army of people to do some of this stuff too. surface viable particle samplings in dynamic conditions throughout the day in the SEC, on carts, on door handles, on the place where you touch to open the door, if that's one of the cases, the pass-through area. As you can see, this is even above and beyond Category 3 standards for environmental monitoring. Let's look at garbing qualification. So talk about even the material of garb. Let's look at it from that perspective. In all essence, in all honesty, we mostly in a 503A are wearing snap smocks, just you put them on as little gowns. But we're talking about this needs to be probably a membrane coverall, which is 10,000 times more effective at particle containment than a standard non-shedding snap smock. So material of garbing, again, these are just little ideas of things that we're trying to look at. What does USP 797 say about competency of garbing? Well, we do a glove fingertip thumb sample, right? Well, if you look at what this may look like, we're talking enhanced garbing competency sampling. Yes, we're going to include that final glove fingertip thumb sampling after donning all of your garb, but then you look at plating the actual garb itself because you've had to put it on. Did you touch it in the wrong place? So looking at plating chest, shoulders, sleeves, thighs, mask, goggles, and hood, all in addition to the glove fingertip thumb sampling. Looking at was I able to garb without contaminating the garb to bring it into the classified spaces. Garbing competency and qualifications, something not easy to do. And finally, compounding qualification. We're talking media fill process simulations here. Aseptic process qualification could look like something just in the qualification of the process itself, 3 consecutive process simulations to pass, and then requalification every 6 months. And we're looking at the maximum batch yield with 0 failures of all of this. So if you're making 500 of something in a batch, you would have to do that 3 times without contaminating a single one, doing a process simulation with media, growth media. That's just the process in and of itself. Is my process work? That's not qualifying each individual person. Then you move to personnel qualification, which is basically the same thing. Initial 3 consecutive process simulations, but in this sense, their minimum is doing 20% of the batch that they would normally do or of a batch. Again, we're looking for 0 failures. It's not for the faint of heart. It's very rigorous. Automation, again, a great tool. I think, repeatability and reproducibility of automation lend itself better to this type of process validation. And then I would even look at aseptic processing plus terminal sterilization if I was looking at doing this, because USP 797 is clear that terminal sterilization is the preferred method of sterilization unless the specific CSP or container closure cannot tolerate the terminal sterilization. So we can aseptically process and then terminally sterilize. This is what it might take to do some type of process validation on this. So remember when I stated earlier that process validation is complex? It is, right? We kind of see that. For compounders that do not have any experience with this, I encourage you to grab a hold of USP 1223s and see that process and the vigors of it. What's interesting is this is the hard pill to swallow. I really do think it is unlikely that most of us have the experience or the knowledge to perform true process validation independently, and should consider, other subject matter experts to verify a validation plan. It's very complex. So back to question number 1. What is true concerning the application of the utilization of the alternative technology statement in section 1 of USP 797? The statement is meant to encourage innovation and flexibility with regards to emerging technologies. In question number 2, process validation can be characterized as an integral part of CGMP and an evolving concept in USP 797. It is not explicitly defined on the how to do it by the Food, Drug, and Cosmetic Act or the FDA. Moving on to USP compounded preparations monographs, demystifying this, kind of looking at it from the practical perspective. In our questions, USP compounded preparation monographs are recommendations for how to prepare, articulate the quality expectations for a medicine, including its identity, strength, purity, and performance. And a compounded preparation monograph is one of 3 of the USP monograph categories. True or false? The USP Compounding Expert Committee is responsible for developing and revising compounded preparation monographs. So what are USP monographs? Well, they are not merely recommendations. They truly are expectations and standards to ensure quality. These recipes date all the way back. This idea dates all the way back to the inception of the United States Pharmacopoeia in the 1800s. So what are the 3 USP categories for monographs? There's product monographs. Those are the ones that are commercially manufactured. So normal saline, D5 bags, frozen vancomycin CSP. They really are conventionally manufactured drug products. The bulk substances or other ingredients that provide standards for identity, quality, purity, and strength. We're really talking about here active pharmaceutical ingredients or API that are used in either product monographs or compounded preparations monographs. And then compounded preparations monographs, what we're going to talk about today as compounders, contain formulations for human and animal patients. And they are specific. They are specific preparations to assist practitioners in compounding these formulations. Examples, morphine sulfate compounded injection is an example, or even from a non-sterile perspective, an atenolol compounded oral solution would be another one. So the compounded preparation monographs by the numbers. Currently, there are 237 compounded preparation monographs, but there's only 18 that are sterile. Of that 18, 2 are ophthalmic, there's 2 irrigations, there's 11 human injection preparations, and 3 are confined to veterinary. So 18 out of 237 is not a lot. Unfortunately, there is only 1 sterile to sterile compounded preparation monograph. This is something we need to change somehow. I don't know how we're going to do that, but this is something we do need to change. It would be nice to have some more of these. So let's look at what the parts of a compounded preparation monograph, or at least the format of them. It starts with formulas. It has directions. It talks about the assay or the stability indicating method, tests, packaging and storage, and goes all the way down to the standards. Obviously, the important part there for us is beyond use dates, is understanding that. That is one of the reasons a lot of people would like to use compounded preparations monographs, is because of the extended beyond use dates. So where do they come from? Well, guess what? The USP Compounding Expert Committee is charged with creation. They do this based on the need in the public, or what we need to look at. The other part would be monograph donation to USP. That is, there have been a number of mostly non-sterile stability indicating studies that have been donated to USP for inclusion in the USPNF. And so that is really important. That is a way to get those into USP. Think of this, really think of a compounded preparation monograph as a recipe. That's really all it is. I've looked at this as, because I've looked at compounded monographs in the past a lot, they could be the perfect evidence-based compounding reference. It's really done with the rigors of USP standards, and those USP standards are really second to none. You know, can this be the ultimate stability indicating study? I think so. I only wish there were more. I wish there were more USP compounded preparation monographs. We would be able to use them without having to conduct our own studies. The future is going to lie in some of these and some processes that a number of people are working on to see if we can donate some more of these, or even figure out ways that USP can do a few more of these monographs for us. So let's dive into the only sterile to sterile compounded preparation monograph. It is a fentanyl citrate and ropivacaine hydrochloride compounded injection. The nomenclature or the name is descriptive in the sense that the compounded preparation monograph will always contain the word compounded in their name. If it doesn't have compounded next to it, it is then a product monograph. From there, the definition walks the compounder through the directions, the resulting concentration to make 1 unit of the product. So it really is a recipe to make 1. For this particular sterile to sterile product, note that the manufacturer is listed, Fresenius Kabi, on the fentanyl and the ropivacaine. That is important because in the case of a compounded preparation monograph, that manufacturer must be used. The second part, or the going down looking at the recipe of the monograph, is the assay or the stability indicating study. The assay is part of the monograph that determine the concentration or potency and acceptance criteria that the reference lab used to determine the values to be applied in the acceptance criteria. This is just a screenshot and really just a snippet of the assay for the fentanyl and ropivacaine compounded injection, but it literally goes through very, very detailed chemical and physical analysis that makes my head hurt. Brings me back to all those days in pharmacy school. Now, let's get to the part that means something more to the compounder, the things that we have a little bit more control over in looking at it. So specific tests, a very important part. It's where the rubber meets the road, so to speak. These are the tests that must be performed and passed on samples from each batch before the batch can be released. Failure of any element of these renders the batch as non-acceptable and it can't be used. And this is a must. So for this particular monograph, we need to test it for pH, and we need to do a sterility test, and we need to meet the requirements of 788 in particulate matter for injections. And it must meet the requirements of having very little particulate. We send these off to reference labs. The additional requirement section is also very important. It contains the practical instructions for the container closure. You know, what type of container closure? This one says package in a light resistant single dose container. It gives us protect from light declarations, storage conditions, beyond use date, and what is to be included on the label. This example, so this fentanyl ropivacaine compounded injection has a 90-day room temperature dating. You can see why we'd love to be able to use and have more of these compounded preparations monographs, because some of them are 90 days, 120 days. And when we're looking at the BUDs of USP in the limits of the chapter, for this situation in category 2, we're looking at a 4-day beyond use date based on this, if we do sterility testing, we get a little bit more, but we're only looking at 30 days, right? Sterility testing in category 2, 90 days room temperature with the stability studies. Now let's look at a compounded preparation monograph using API, sodium phosphates, compounded injection. As you can see, this one is a measurement. We're starting to weigh things out now. Again, my head's starting to hurt a little bit. We're weighing ingredients. We're talking about dissolving the powder and the active pharmaceutical ingredient into a solution, and then filtering the solution to remove any particulate matter. In this compounded monograph, we are actually sterilizing by autoclave. So that is very important. That must be done. That's how we achieve sterility, is using the autoclave. So this is going to be in a glass vial. Here are the specific tests: pH, sterility tests, and it must pass bacterial endotoxins, so USP 85, because we're using active pharmaceutical ingredient. Remember, bacterial endotoxin test is not necessary for a sterile to sterile type product. And then the final preparation is packaged as a single dose glass container with the resulting BUD of 120 days at room temperature. And if you're familiar with the labeling of a commercially prepared vial, you recall that the vial is labeled just like it's being asked to be labeled here: 4 milliequivalents per mil of sodium and 3 millimoles per ml of phosphorus, just like the labeling requirement from the commercially manufactured product. Here we are, a great exercise or an example of active pharmaceutical ingredient using a compounded preparation monograph. So aside from the intricacies of the assay portion, so that stability indicating study section, it definitely would be, I hope you're not really intimidated, and it would be definitely good if we could get more of these sterile CPMs using sterile to sterile ingredients. I know this is something that USP would welcome in and of itself. So when you're utilizing these compounded preparation monographs, we must use the components exactly as they're described in the definition. And I cannot overemphasize the word exactly. The instructions for compounding the preparation and the definition must be followed as written. Compound the exact concentration in the definition or concentrations that are in a range of a bracketed monograph. For example, if the monograph is listed as 1 milligram to 4 milligrams per ml, you can compound within that bracketed range. Our example of the ropivacaine was fentanyl was not a bracketed study. It was a specific concentration. The volume as described in the compounded preparation monograph is typically 1 unit. Additional quantities are allowed provided the proportion remains consistent with the directions. And the specific tests listed on the compounded preparations monograph must be performed, and the results received on a representative sample of the batch prior to release. The ability to release ahead of time before this information comes back means that you are subjecting yourselves to the limits of USP 797 in section 13. A word of caution: there is 0 deviation from the compounded preparation monograph. For example, if you're compounding this fentanyl and ropivacaine and you use a different manufacturer of either the fentanyl or the ropivacaine, which is listed as Fresenius Kabi, you can, and you're only permitted to assign the BUD limits in the chapter. For the API, think salts, think the different salts. Think sodium phosphate has a dibasic and a monobasic, and the multitude of choices that could be available. Choosing one that is not in the monograph would make that monograph invalid. Most importantly, any omission of any of the required specific tests or additional requirements would also necessitate reverting to the BUD chapter limits. So this compounded preparation monograph is not a recommendation, it is not a suggestion, it is not grandma's recipe, a pinch of this, we're going to add a little bit more salt here. We follow these exactly, or we are invalidating the monograph in and of itself. So let's look at question number 3. A USP compounded preparation monograph, are they recommendations to prepare a compound and its associated beyond use state? Do they articulate the quality expectations for a medicine, including its identity, strength, purity, and performance? Are they is it one of 3 categories of the monographs? And it is B and C. Remember, they are not recommendations for how to prepare a compound. They are very specific, and its associated beyond use date is dependent on preparing it exactly as it is written. And true or false, the USP Compounding Expert Committee is responsible for developing and revising compounded preparations monographs, and that is true. So let's shift gears here again and talk about something that I'm very passionate about, and that is protect from light factors. Question: most protect from light statements included on package inserts or prescribing information refer to either short-term storage, administration, long-term storage, dilution and preparation, or all of the above. And true or false, a change in color of a CSP always means that photodegradation has occurred and the CSP should be discarded. So in my practice, protect from light considerations of sterile products is a very common inquiry. According to ISMP, actually the phrase protect from light is poorly defined in prescribing information and in many drug references. Inconsistency exists in the understanding of what protect from light means and the necessary measures that should be taken during the various phases of the medication use process. I'm going to have to say, I wholeheartedly agree with ISMP on this. So there's a bunch of things to consider. We really need to understand how we interpret this information and move forward and apply it in our practice. A few definitions: photostability being the ability to be resistant to change when exposed to radiant energy, especially light. So photostability studies are what manufacturers look at when they're designing drugs and drug packaging. Even some stability indicating studies for long-term storage of compounded preparations will add a photostability study if needed. So photodegradation is the breakdown of medications induced by light, not only just injectables, but oral solids, oral solutions, things like that. And then protect from light. What is that? Well, that's the physical process of shielding a drug or packaging that blocks out the light. And it really is dependent on many factors, where it is in the process and the life cycle, and what is the rate of degradation that could be occurring. So looking at the objectives of conducting photostability studies is to evaluate the overall photosensitivity of the pharmaceuticals for the development and validation purposes, and to provide the necessary information for handling, packaging, and labeling. The studies are basically determining how the medication reacts to light and how the results of the study can be extrapolated to the life cycle of a particular medication. We're looking at this spectrum of what the photostability studies are trying to show us and how then we in the pharmacy take care of those protect from light situations. So here's a depiction of the life cycle of a medication, and you can see from the manufacturing to waste, so and everything in between. The portion of USP 797 beyond use dating is applied from the point of reconstitution through the preparation, and even a possible short-term storage in the pharmacy or automated dispensing cabinet needs to be considered. Additionally, there could be short-term storage during a stock process. Let's say we get a tray in, or we're taking medications out of boxes and things like that. All of those situations may warrant the understanding of the photostability of the medication and the packaging and the resulting process of even the compounded sterile product. So understanding kind of the life cycle is where we're going to talk about what these measures look like and how we want to apply them. I've included this slide here just to give you an idea of some light-sensitive chemical reactions of some medications. Lots of big words here, lots of things that make my head hurt. But you can see that the vitamins B2 and B12, nifedipine goes through a photodehydrogenation, and atrienam photoisomerization. So some very common drugs here that go through some chemical reactions. So I think you're going to understand this in a second, but not all photoreactions are degradation. I think we've all seen this, that cefazolin bag that has either been made, sat around for a little while, gone up to the floor and come back, but it's not degraded. We know that cefazolin does change color. There is a chemical reaction that goes on to change color, a physical reaction, but the drug is actually not go through degradation, which is very interesting. So just remember, not all photoreactions are photodegradation. Now that we've defined photostability, kind of talked about its importance and understanding its effects and how our CSPs are prepared and stored, let's dive into the practical aspect of where do I find information on protect from light recommendations? Well, unfortunately, most protect from light declarations are found in the package insert, and that information typically makes its way into tertiary references. So why do I say unfortunately? Is because the manufacturer is most likely stating the protect from light recommendations for long-term storage. And as you would expect, we have a tendency to take those words, protect from light, wherever we see them, and extrapolate them, sometimes erroneously, to any of the protect from light declarations all the way to patient administration. So we need to look at it in the essence of the life cycle. So I'm not saying that all package information or package inserts are incorrectly applied, but there seems to be only a few package inserts that clearly define the CSP protect from light recommendations. My favorite resource is the Light Sensitive Injectable Prescription Drugs 2022 article published in Hospital Pharmacy. It is a peer-reviewed compilation of the different sources, but also presented in a format that is in line with the life cycle of a CSP. It is an invaluable resource in my practice, and I will freely admit it is my go-to guide when I'm looking at these, because in my practice, this comes up a lot. So let's take an example of a medication that we're looking at, and with including the snippet from the article. Leucovorin injection clearly states in the package insert, protect from light. However, it has been determined that protect from light measures are only required for long-term storage and not the other parts of the life cycle. How many of your practices are applying a protect from light measure to leucovorin, either in syringes or in compounded bags for administration? I would bet a number of you are shaking your head yes. Let's jump to another example, sodium nitroprusside. This is an excellent example of a package insert and enclosed contents making it clear that a compounded sterile product must be protected from light from storage through administration to the patient. As you see, they even include their own protect from light measures in the box. Furthermore, and it is kind of my bellwether or the thing I look at the most, I do like the fact that the injectable is supplied in an amber vial in this perspective. That does not mean that all amber vial medications require protect from light measures, but some of the ones that are manufactured and packaged in clear glass vials, and then we think we need to protect them from light, becomes a point of confusion amongst our staff and amongst looking up the different information that we find in the literature. Assuming you're utilizing the right material, these medications and these plastic materials that block out ultraviolet light, you can use all the way from long-term storage all the way through administration. But I want to point out, not all of these bags or materials are created equal. We want to look for light transmission not exceeding 10% at any wavelength from the 290 nanometers all the way through 450 nanometers. And that's important for the ultraviolet part of that. Bags that are transparent but amber, you know, the ones that you can actually see the label through, they're just kind of a slight brown, are probably not sufficient in blocking the correct spectrum of the wavelength. In this case, I would think we're not applying appropriate protect from light measures, which could lead to as much photodegradation as an unprotected compounded sterile product, depending on the medication. I think what I'm trying to say here is looking at what you are using for protect from light and to choose wisely. Make sure you're making an informed decision on the protect from light measures that you are using. So the question then becomes sometimes is, why not employ protect from light measures all the time across the board at all stages of the life cycle? So here's one of those bags that may not be protecting from light because it's too light. I don't know, but this was the example because I wanted you to be able to see through the bag and see that there is a bag inside a bag being protected from light. So why not employ these light measures? Well, most injectable medications photodegrade so slowly that they only need to be protected during long-term storage and not during compounding or administration. Again, we're talking about protecting everything, not the ones that we know for sure need protect from light measures. And protecting every injectable medication at all stages of the life cycle probably consumes excessive resources. And we're talking about time, buying more materials, the process of putting them in there, and then the confusion that it causes if a protect from light bag is omitted from maybe that process. Well, here's a novel idea, and ISMP in 2024 nailed it. They're saying for drugs that require protection from light, the FDA should consider revising labeling to require more detailed information on the duration of light exposure that may result in medication degradation to ensure appropriate light protection of medications during the entire medication use process. So ISMP has gone out there on a limb. Hopefully the FDA will start looking at that. Excuse me. So how to simplify your protect from light approach? Evaluate a standardized approach to protect from light processes and your labeling from your EHR. Continue to assess and make determinations based on the specific life cycle stage. Based on where the protect from light statement is found, sometimes out on the outer packaging, could determine what that means. If it's on the outer packaging, does that mean long-term storage? Maybe, but you have to verify that. Light sensitive injectable article is the go-to, in my opinion. It is a great resource, and it is actually hyperlinked in the slide if you've downloaded them. And then finally, I do think that protect from light precautions are probably overused and unfounded in a lot of cases. So look at that in your organization and try to figure out what some of those places are. So final questions. Most protect from light statements included in package inserts or prescribing information refer to long-term storage. And true or false, the change in color always means that photodegradation has occurred and the CSP should be discarded. No, that is false. Look at that. Error on my part. Should not be discarded because remember we talked about the cefazolin example. So, are there any questions in the chat? Let's see. And here is the CE credit slide for you to claim your CE.
Thank you, George. This is a fantastic session. As a reminder, attendees can still submit questions using the questions chat. And we do have quite a few questions in there, George.
We do have quite a few questions. How to get an up-to-date list of the compounded monographs. That is available on the USP website. If you just Google, actually is how I found it. I know a member of the compounding expert committee didn't even know it was there. There's a PDF of all the USP compounded on their website. If you Google USP compounding preparations monographs, it'll take you to the page that has the hyperlink to download a PDF. Is my simplified process validation or repeatability? How many times must be able to repeat the results to confirm validation? That's a great question. And a lot of times the FDA looks at this magic number and it's entailed in a lot of their different articles, but that number becomes 3. Now, I think there's ways to prove without doing 3. The FDA likes the number 3. So we have seen a number of 483s at different outsourcing facilities where they wanted to see process validation completed 3 times. And I think with that, I will take these questions and I will answer them offline because I know everybody's got things to do, but thank you ARL and thank you everybody for your participation. And I look forward to seeing you around again at another time.
Wonderful. Thank you again, George. This was a fantastic session. As a reminder to the attendees to receive CE credit, complete the evaluation link on the last slide of the handout. And we will also be sending a follow-up email 1 hour after today's session that will have a download of the slides that you saw today during the session, and it will also have the evaluation link. You have until November the 29th to obtain your CE credit. Thank you very much and have a wonderful afternoon.